Howdy, today we'll be presenting the cell-mediated immunity, presented by Tanya, Rowan, and Emiliano. T-cell effector functions. T-cells play an important role in cell-mediated immunity, which protects the body against microbes that live inside the host. There are two main types involved in this process, helper T-cells and cytotoxic T-cells. Helper T-cells, or CD4, mainly coordinate the immune response. They release cytokines that activate macrophages and recruit other immune cells to help eliminate pathogens, while in cytotoxic T-cells, or CD8, they directly kill infected cells. They recognize infected cells displaying microbial antigens and destroy them, which remove the source of the infection and help prevent the pathogen from spreading from others. Activation steps. This diagram shows the main steps involved in the activations of T lymphocytes. First, a naive T cell recognizes an antigen presented by an antigen-presenting cell. This recognition begins the activation process. Next, the T cell undergoes colonial expansion, which means it rapidly divides to produce many identical T cells that recognize the same antigen. Cytokines like interleukin-2 help stimulate this proliferation. As shown in this diagram, T cells can differentiate into effector T cells that help eliminate the infection and memory T cells that remain in the body and allow for a faster immune response if the same pathogen appears again. Activation signals. First, the T cell receptor recognizes a peptide antigen presented on the MHC molecule by an antigen-presenting cell. CD4 or CD8 co-receptors help strengthen this interaction. The second signal is co-stimulation, where B7 molecules on the APC bind to the CD828 receptors on the T cell. When APCs are activated by microbes, they increase co-stimulator expression and release cytokines. This leads to the interleukin-2 production, which causes T cell proliferation and differentiation. Without the co-stimulatory signal, the T cells does not respond and becomes tolerant.
This slide shows the main receptors and ligands involved in activating CD4 plus T lymphocytes when it interacts with an antigen-presenting cell. First, the T cell receptor, or TCR, recognizes a specific peptide that is being presented on MHC class II. CD4 acts as a co-receptor by binding to class II MHC as well, which helps tracing the interactions and support signaling. CD3 and the zeta chain are part of the TCR complex and are responsible for signal transduction inside the cell after antigen recognition. For full T cell activation, co-stimulation is also needed. CD28 on the T cell binds to B71 or B72 on the antigen presenting cell. This provides the second signal that promotes activation. In contrast, CTLA4 and PD1 are inhibitory receptors. CTLA4 can also bind to B7 molecules, while PD1 binds to PD-L1 or PD-L2, and these interactions help turn down or limit the T cell responses. Lastly, LFA1 binds to ICAM, which helps with adhesion and stabilizes the contact between the T cell and the antigen presenting cell. So overall, this slide shows that T cell activation depends on the antigen recognition, co-stimulation, inhibitory regulation, and adhesion. This slide summarizes the major effector mechanism of T cell mediated immunity by comparing the roles of CD4 plus helper T cells and CD8 positive cytotoxic T cells. On the left side, the slide shows CD4 positive effector T cells responding to microbes that have been ingested by phagocytes and are located inside the vesicles. Two important subsets are shown here: Th1 cells and Th17 cells. These helper T cells do not usually kill microbes directly. Instead, they secrete cytokines, which are signaling molecules that activate the other immune cells. Th1 cells mainly activate macrophages, which improves the macrophage ability to destroy the microbes they have already engulfed. This is especially important for pathogens that can survive inside phagocytes. Th17 cells, on the other hand, promote inflammation. This inflammatory response recruits additional leukocytes such as neutrophils and monocytes to the site of infection, helping the body kill microbes more effectively. On the right side, this slide shows CD8 positive cytotoxic T cells, lymphocyte or CTLs responding to infected cells that contain microbes in their cytoplasm. Unlike CD4 helper T cells, CD8 T cells directly kill the infected cell. This is important for eliminating cells infected by viruses or other intracellular microbes. So overall, this slide highlights the two main strategies of T cell mediated immunity. CD4 T cells coordinate immune responses through cytokine secretion, while CD8 plus T cells directly destroy infected cells.
There are four main subsets of CD4 cells. There are T helper type 1 cells, which secrete mainly interferon gamma, stimulating the classical pathway of macrophage activation. They provide defense against intracellular pathogens and can result in chronic inflammation and autoimmunity if unregulated. T helper type 2 cells secrete interleukin 4, 5, and 13, primarily targeting eosinophils, mast cells, and the alternative pathway of macrophage activation. They provide defense against helminth infections and aid in tissue repair and fibrosis. T helper 17 cells secrete interleukin 17 and 22, recruiting and activating neutrophils to phagocytize extracellular bacteria and fungi. Last but not least, there are the follicular T helper cells, which secrete interleukin 21 in addition to interferon gamma and interleukin 4, and reside in the follicles of secondary lymphoid organs in order to assist B cell production of antibodies to target extracellular pathogens. Antigen recognition by the binding of MHC class II on the macrophages and the TCR on Th1 cell occurs, stimulating the classical pathway of macrophage activation. The Th1 cells secrete interferon gamma, and at the same time, the CD40 ligand and CD40 receptor bind and provide the necessary second signal for macrophage activation. This results in increased macrophage production of reactive oxygen species and nitric oxide in order to kill engulf bacteria. Sensitive to antigens also increases as MHC and co-stimulator expression increase, and cytokine release causes inflammation. Interleukin 4 and 13, secreted by Th2 cells, stimulate M2 macrophages. This alternative activation results in the secretion of interleukin 10 and transforming growth factor beta by the activated macrophage, providing inflammatory effects in tissue repair, which could lead to fibrosis. If M2 activation overtakes M1 activation before the infection is completely eliminated, it can lead to prolonged and more severe cases of disease as recovery starts to occur before the infection is actually eliminated. In order to kill infected cells, CD8 cytotoxic T lymphocytes must recognize an antigen through their TCR binding to the MHC class I molecule on the infected cell. Leukocyte function-associated antigen binds to intracellular adhesion molecule 1 in order to stabilize the binding. Once attached, perforin is secreted by the T cell, poking holes in the infected cell in order for granzymes to go in and activate apoptosis of the infected cell.
Here are the member roles and their tasks. These are the resources we use. We use the basic immunology textbook and BioRender. Thanks and Giga.